So hi, I'm Georgia, and I'll speak to you today about my research in modelling clearance and proteopathy in Alzheimer's disease using network models. I work under the supervision of Professor Alan Gorielli at Oxford and also Professor Marie Rognes at Simula, and I also work with Dr. Travis Thompson and Dr. Hadrian Oliveri at Oxford. Alzheimer's is a predominant disease, which is becoming an increasing problem worldwide due to increased life expectancy, but it is surprisingly poorly understood. In Oxford, we use mathematical models of neurodegeneration to better understand the fundamental mechanisms which underpin the disease, and this brings us closer to answering the key questions at the forefront of Alzheimer's research. Neurodegenerative diseases are characterized by the accumulation and propagation of toxic proteins in the brain. For Alzheimer's disease, these trademark pathogenic proteins are amyloid beta and tau proteins, which are pictured here, where tau tangles are inside the neurons in blue and amyloid beta plaques are in brown, forming plaques outside the neurons. The characteristic spreading patterns of these proteins through the brain are predictable, and there's a well-established staging of the disease. So this is one element that makes Alzheimer's so unique in comparison to other unpredictable diseases. We understand how Alzheimer's progresses over a person's lifetime, and this is useful for mathematical validation. So this is how we define the disease, but the mechanisms driving the disease are poorly understood, and there are many open questions. For example, why do we see such distinct spreading patterns? What dictates these spreading patterns? Why does the disease progress so slowly? So it takes decades before you see the infamous symptoms of Alzheimer's, such as forgetfulness. Can we interfere in the spreading? Because at present there's no cure. And how do the driving factors of the disease interact? So then a big question is, what are the driving factors of the disease? And this is what informs the structure of our mathematical model. So two main driving factors of Alzheimer's are thought to be protein propagation and aggregation and clearance pathways. Here on the left, we see scans of healthy and unhealthy brains where increased protein concentration regions, and proteins spread through the brain in a prion-like manner, where toxic proteins come into contact with healthy proteins and act as a template upon which healthy proteins misfold and also become toxic. In a healthy brain on the top here, you can see that toxic proteins occur naturally, but they're cleared from the brain through multiple interconnected clearance pathways. So the rest of the body has a lymphatic system, which acts as a sewage system for toxic waste, but the brain doesn't have a lymphatic system. Instead, it flushes toxic proteins out through the blood or cerebrospinal fluid mediated clearance or by degradation. But it's thought that an imbalance between protein production and clearance leads to a cascade of proteins through the connectome, which results in Alzheimer's disease as shown on the bottom left here, in an unhealthy brain where toxic proteins have invaded and saturated more regions of the brain. Interestingly, the accumulation of toxic proteins damages clearance pathways, and reduced clearance leads to the accumulation of more toxic proteins. So there's a toxic loop of damage here, which is key to our model. At present, research into Alzheimer's is focused on either protein propagation, aggregation, or clearance. So our aim is to bridge the gap between the two and combine them both into one mathematical model, which may be quite a big missing piece in our understanding of Alzheimer's disease. Looking at the staging patterns of Alzheimer's, we can see that it's clearly not a simple extracellular diffusion pattern of toxic proteins through the brain. Moreover, imaging studies have shown toxic proteins to move along axonal fiber tracts in the brain rather than by proximity. Diffusion tensor imaging, like this one shown here, shows molecules moving along axons in the brain. So by looking to DTI images, we can gain insight into the location of these axonal highways, which proteins essentially hijack in the brain. So using insights from DTI images on the structure of axons in the brain and using simple reaction diffusion models, Wickermeyer and Fenari et al. Take continuum and network approaches to modeling Alzheimer's disease. The continuum models, with results shown in the middle here, are anisotropic, and they use some notion of axonal directionality with both extracellular and axonal diffusion. But the network models really explode this notion that the network is what transports proteins by restricting transport to being along axonal fibers. Now, comparing the continuum and network models to clinical observations on the top row, you can see that network models capture the characteristic spreading of Alzheimer's in the same way that continuum models do, but at a fraction of the computational cost. So on the bottom row here, you're seeing the progression of Alzheimer's, which is simulated for over 40 years of a person's life. And this took less than 7 seconds to run on a standard laptop computer. So based on this work, we proceed by taking the network modeling approach. So here's our brain network on which we will model the spread of Alzheimer's disease. And here, the nodes are predefined regions of interest, and the edges have weights proportional to the number of axonal fibers between regions. We describe transport of toxic proteins on the network using the graph Laplacian, which is defined here on the left. And this is the analog of the continuum Laplacian operator for diffusion. Previous authors have modeled the spatiotemporal spreading of toxic proteins well with simple reaction diffusion equations, such as the Fisher-KPP model. So here, the Fisher model considers a conversion of healthy to toxic proteins at a constant rate alpha. And the resulting biomarker curves on the right show the toxic concentration in each lobe progressing over time. And it shows a sequence of invasion which reflects imaging studies well. But the progression of Alzheimer's here is directed only by the topology of the brain network. There's no clearance or aggregation kinetics in this model. So how can we overcome these limitations and bring a dynamic clearance into the model while still keeping the model simple? Well, first, let's look at some more complex models. So in vivo, proteins aggregate by complex protein kinetics. And here's a picture of those mechanisms where toxic proteins are in red and healthy proteins are in blue. We consider multiple effects for the formation of aggregates in our system of different sizes with rate constants Ki, while transfer of mass to and from the system are represented by constants gamma and lambda I. And we can model this using the Smolchowski model, and this is widely used in statistical physics to model the number density of particles of different sizes, for example, in modeling aerosols. We won't go into detail on the equations here, but we note that unlike the Fisher model, this considers a bulk constant clearance in the form of these constant clearance rates, lambda 1 and lambda 2. This gives rise to a really important result, which is that there's a transcritical bifurcation in the system, and it reveals a critical clearance rate, lambda crit, and this is a protein-specific constant. The critical clearance is important because it marks the rate above which aggregation is negligible, and if clearance in a region drops to below critical, toxic proteins will overrun and saturate that region. The critical clearance is a quantity which we'll take forward in our model, which we'll detail in the next slide. So this is our model. It's of similar form to the Fisher model, but it considers a dynamic clearance and incorporates this critical clearance rate, lambda crit. Here, P denotes the toxic concentration. It's modeled by a reaction diffusion equation for this prion-like spreading and clearance of toxic proteins. And the first term here uses the graph Laplacian to model diffusion across the network, while the second two terms describe the local dynamics of interaction between toxic protein concentration and clearance and the saturation of a region in toxic proteins. This is coupled to a simple decay law for clearance lambda, and the third equation describes the evolution of damage Q, which is correlated with downstream effects of Alzheimer's, such as atrophy and functional decline. So here we've captured this toxic loop of damage, which we described in the background slide, where toxic protein concentration increases damage, which inhibits clearance, which an inhibited clearance actually increases toxic protein concentration. So this model is different to previous models because now clearance is a dynamic variable. And we'll see that this gives rise to much richer dynamics in the system. And to proceed in our analysis of the model, we ignore diffusion and we analyze the local dynamics in a single node. By ignoring diffusion in this way and restricting our attention to the single node case, we reveal some interesting patient states. And these patient states correspond to fixed points A and B in the node, which correspond to either zero toxic concentration or a region which is fully invaded, fully saturated in toxic proteins. And the stability of these states is dependent on the clearance lambda. So here's a phase plane for the system with toxic protein concentration on the y-axis and clearance on the x-axis. And using the phase plane, we can establish a closed formula for a critical toxic seed, which will drive the clearance in a node to below critical. The critical toxic seed therefore represents the total toxic burden that a region can withstand either incrementally or all at once before transitioning from a healthy state to a disease state. So here we see that if we seed an amount less than this critical toxic seed, PC, then the node tends to a healthy state, which is actually a line of fixed points along the null line at P equals 0. And if we seed above PC, the clearance is driven to below critical and the toxic proteins will saturate the region, as shown here. We also display biomarker curves for clearance in blue, toxic concentration in red, and damage in black. And we see in the top middle here that while clearance does drop, there is damage to clearance mechanisms. The toxic concentration is cleared to 0 before the clearance, it drops below critical, and so the brain remains in this healthy state. Now we extend to an entire network. Using this concept of a critical toxic load, we seed in the entorhinal cortex, which is where tau proteins are first found in Alzheimer's. And our model starts with a healthy brain that initially has sufficient clearance to eliminate small amounts of toxic proteins. However, as the toxic load increases, the clearance systems become increasingly damaged and the brain is subject to full invasion. We also display the evolution of biomarker curves for clearance and toxic protein in each lobe on the bottom here. And these curves show that the order in which lobes are invaded are in agreement with clinical observation. The brain network here takes much longer to reach full invasion with my model compared to the Fisher model. So linking this back to one of the questions at the beginning, why does a disease spread so slowly? This could be why. Clearance essentially puts walls up between regions in the brain, and proteins need to hack away at this wall to drive clearance to below critical so that they can go on to invade the region. The computational results show that an increase in the level of healthy initial clearance delays the onset and progression of neurodegeneration. We see a non-linearly increasing relationship between the initial clearance and onset time across relations, which means that improved brain clearance, especially before neurodegenerative onset, may have significant brain benefits to brain health. So far in this talk, we've considered a uniform initial clearance landscape in our model. But in real life, does the landscape of regionally varying clearance play a role? Perhaps some areas of the brain are more resilient than others. So we'll now go on to explore the impact of heterogeneity in clearance on these spreading patterns that we observe in Alzheimer's disease. And we'll do this through 2 case studies. So now we're touching on another question from the introduction, which is what dictates the spreading patterns that we observe in Alzheimer's disease? The first such study concerns Alzheimer's subtypes. So there's been a considerable number of recent studies which examine different subtypes of Alzheimer's progression. And we'll now see if clearance could be a factor in creating pathways for Alzheimer's subtypes. The subtypes are classified using the ratio of toxic tau accumulation in the hippocampal region, shown in orange here, and the association cortices highlighted in pink. In hippocampal sparing Alzheimer's, the hippocampal region is less saturated. In typical Alzheimer's, these regions are comparable. And in limbic predominant, the hippocampal region is more saturated. So we engineer initial clearance maps, which we apply to our model, and we recover the different Alzheimer's subtypes as shown here in our simulations. And interestingly, these results recover the clinical observation that the simulated onset time of the hippocampal sparing variant reaches onset earliest, followed by typical Alzheimer's, and then followed by limbic predominant Alzheimer's disease. Therefore, a key finding of my work is that the coupling between the spreading of toxic proteins and regional brain clearance not only influences the trajectory of Alzheimer's disease, but it could also provide a potential window into Alzheimer's subtypes. Now, in the next case study, instead of using purely theoretically engineered initial clearance maps, we'll derive initial clearance maps from state-of-the-art human clearance data, which was provided by our collaborators in Norway. And this is derived from a landmark trace study in humans, which studies glymphatic clearance in a rested and a sleep-deprived cohort, where glymphatic clearance is a type of cerebrospinal fluid-mediated clearance. So why are we interested in sleep? Well, it's thought that clearance rates increase during sleep, and so sleep deprivation has harmful effects on clearance. Moreover, using our model, we can bridge the gap between glymphatic clearance hypotheses and actual Alzheimer's progression using this human clearance data. So here the tracer used mimics tau proteins being cleared from the brain, and the MRI acquisitions, which are shown across the screen here, took place over the course of 1 month. We see the tracer intensity peak at around 6 to 24 hours, and we're interested in the clearance of the tracer from this point down to the baseline at 4 weeks. So here's the tracer intensities for the sleep-deprived cohort in red and the reference cohort in blue. And from the peak intensity in the tracer, we fit an exponential model for clearance of toxic proteins in each region of interest. And in this way, we extract initial clearance values in each region and produce an initial clearance map across the entire connectome for the reference cohort and the sleep-deprived cohort. And we apply these initial clearance maps as initial conditions in our model. We see statistically significant differences between these initial clearance maps in both reference and sleep-deprived cohort. And we also see statistically significant differences in the resulting toxic protein pathologies. For example, as shown here, Alzheimer's spreads faster in the sleep-deprived cohort, which is on the right-hand side, and also along a different trajectory when we compare it to the reference cohort on the left. So this supports the suspicion that sleep deprivation may be a potential contributing factor for Alzheimer's disease. And it also shows that heterogeneity in clearance and different initial clearance maps leads to different trajectories of Alzheimer's disease. Focusing on the results from the reference cohort, pictured on the top here for a well-rested patient, and focusing on the final time point in particular, we'll compare this to actual patient data taken from the ADNI database. So here's an SUVR image on the bottom here in green. And you can see that now our simulation better reflects reality compared to previous models, because we see an increased concentration of tau on the left side of the brain, on the top row here, and also in the SUVR image in green on the left, you can see an increased toxic concentration of tau. And this is a feature that we haven't been able to recover before. So it shows that regional clearance could bridge the gap between simple mathematical models and actual patient data. So in conclusion, the clinical community has raised an alarm on the connection between Alzheimer's disease and the brain's inability to clear toxic proteins with age. And we heed this alarm by producing the first mathematical data-driven model which couples clearance and proteopathy. This work reveals that indeed clearance could play an important role in Alzheimer's development. Future work includes extending to other neurodegenerative diseases, running computational drug trials with our software and model, and also exploring this question further: can sleep deprivation bring on Alzheimer's disease? So we have a preprint out on bioRxiv right now, and we're writing another paper on the sleep study, which I touched upon in the previous few slides. I would like to thank you for listening, and I look forward to some interesting discussions.
All right, thank you so much, Georgia. Thank you for the great talk.
Thank you.
So we'll open up for questions from the audience. Are there anyone who'd like to ask a question? You can also type in the chat or just unmute and ask if you'd like to. All right, so I have a question about, oh, there we are, yeah. So this is from Hadi, saying, very nice work. There might be many factors which can change the clearance rate, but how can we link them to Alzheimer's causes?
Yeah, so I agree, there's many things which could potentially change clearance. I think if clearance impacts the trajectory of Alzheimer's disease, and in particular, if a decrease in clearance leads to an accumulation in toxic proteins which characterize the disease, then if we have a decrease in clearance, then this has a knock-on effect to a cause in Alzheimer's disease, I think. And that's what the model seems to suggest. That's what the model does suggest. Other types of dementia. So this can be extended to other types of other neurodegenerative diseases in general. Someone's just asked, sorry, has such work been done for other types of dementia? It can be extended. The reason we focus on Alzheimer's disease is because of the availability of data. And for example, we know certain protein specific parameters such as a critical clearance lambda crit. We have done, well, not me myself, but people in the group have derived specific values for these quantities because of the data available on Alzheimer's disease. But it can be extended to other neurodegenerative diseases because they present in a similar way with a specific seeding site and specific staging of the disease and a specific toxic protein which is responsible for the disease. So it can be extended, yes.
All right, any other last question?
Thank you.
If not, I'd like to ask one quick question about, because when you add the data, oh, here's another.
One thing to observe can be genetic factors changing this clearance, not a question. Oh, thank you.
There's one above as well from Wayne. Thanks a lot for the great talk. I was wondering if your model can guide clinical intervention for optimizing altering the clearance pathway by noting out where it can be most effective.
Yeah, this is definitely something that we're interested in. When I speak about running experimental computational drug trials using our model, I guess that one key area of interest would be if we can somehow increase clearance in this node, would that have a bigger effect on delaying the onset of Alzheimer's disease or even stopping Alzheimer's disease compared to other regions? Yeah, that's definitely a really interesting question to explore in these computational drug trials.
Okay, great. I think that was the time we had